Hi, I'm Dr. Kurt Wohler. The title of this mechanisms video is Lipid Oxidation, Toxic Aldehyde Formation, and Cytochrome C Apoptosis Activation. So this video is sponsored by Integrative Medicine Academy. Integrative Medicine Academy is an online training academy with various mastery courses with different topics on integrative medicine. So to get a full list of our courses, go to integrativemedicineacademy.com, or you can also email us at integrativemedicineacademy@gmail.com. Here is our disclaimer. Understand that this information within this video is for educational purposes only. Now, much of this information actually comes from my course called Mitochondria Mastery Course, which is a very in-depth course about mitochondrial issues from its biochemistry, genetics, the differences between mitochondrial disease and dysfunction, laboratory testing, various interventions, etc. So for more information about the Mitochondria Mastery Course, you can go to mitochondriamasterycourse.com. So let's get into the information here. That we're going to talk about lipid oxidation and how that can be related to mitochondrial problems. So one of the things to understand is that when we generate what are called reactive oxygen species within the body, these oxygen species like superoxide anion or peroxide or the very toxic hydroxyl radical can damage tissue throughout the body. So we know that a very simplistic diagram, something like this free radical taking an electron from a DNA and then damaging the DNA to create a stable molecule is sort of the basic idea of a free radical grabbing electrons from some type of compound. That could be a fat, that could be a protein, that could be some other kind of chemical structure. And then we do have enzymes like superoxide dismutase, catalase, and glutathione peroxidase that help us to neutralize the potential for these oxidative chemicals. So superoxide dismutase converts superoxide radical into hydrogen peroxide, but we've got to have catalase to prevent it from producing hydroxyl radical. And we also have glutathione peroxidase, which helps us convert hydrogen peroxide into water. So it is possible to have deficits of the activity of these various enzymes. And so, for example, if we're forming large amounts of hydroxyl radical, these hydroxyl radicals can attack what are called polyunsaturated fatty acids. These are just fats that could exist in our cell membranes as part of our phospholipids. Now, PUFAs have multiple double bonds which are prone to these oxidative reactions. And the problem is that when a hydroxyl radical is attacking a PUFA, generally what happens downstream is that the radical itself, or excuse me, the fat or the lipid that's being attacked and oxidized becomes its own radical. And so it is a self-propagating problem where it just creates worsening oxidative reactions within the cell. And so it's a real problem, and we need to have good antioxidant reserves to try to buffer against that. Like vitamin E, for example, can help buffer against some aspects of fatty acid oxidation. Now, one of the other problems is that once we have gone through polyunsaturated fatty acid oxidation, we form a couple additional compounds. One is called malondialdehyde, and another is called 4-hydroxynonenal. I'll talk about those here shortly. So this is a little bit more of a detailed discussion of this hydroxyl radical attack of what's called lipid peroxidation on a polyunsaturated fatty acid. And so again, it forms a lipid radical that then itself propagates this lipid oxidation reaction and then finally ends at malondialdehyde and 4-hydroxynonenal. Now, aldehydes are a chemical group that has a carbon double bonded to an oxygen with a hydrogen off of one end. And aldehydes are very reactive in chemical reactions, particularly within our body. And this is a paper that came out a number of years ago looking at aldehyde toxicity and metabolic problems. And the upshot is this, is that excess aldehydes can damage DNA and RNA. They can generate these reactive oxygen species. They can damage proteins. They can damage the mitochondria. They can disrupt membranes in our cells. They can cause lipid oxidation, and all of that can lead to glutathione depletion. So that's going to compromise liver detoxification as well as prevent against excess oxidative reactions within the body. Now, a couple things about malondialdehyde and 4-hydroxynonenal. So malondialdehyde occurs from lipid peroxidation of these polyunsaturated fatty acids, these fatty acids that have multiple double bonds. And so this marker is actually a biomarker of oxidative stress. And the problem is that the malondialdehyde is actually an aldehyde itself. So it can then go off and create its own problems. And one of the things that malondialdehyde will do is it will attack the DNA. So it causes what's called deoxyadenosine and deoxyguanosine DNA adducts. That's just a fancy name for the fact that it causes damage within the DNA. Now, 4-hydroxynonenal occurs because of lipid oxidation as well. Now, this chemical may play some sort of role biologically within the cell in what's called cell transduction signaling. So there can be sort of a normal response that occurs within the cell in the presence of the 4-hydroxynonenal. The problem is that it's been linked in excess amounts to cancer, to Alzheimer's, to cardiovascular problems, diabetes. And very important and interestingly is that it triggers what's called mitochondrial apoptosis with the release of cytochrome C. Now, if you've seen any of my other videos on the mechanism series, I talk about different aspects of mitochondria. Many of my webinars and lectures through Integrative Medicine Academy, we get into this in great detail, particularly in the Mitochondria Mastery course. This is the inner mitochondrial membrane right here. And within the inner mitochondrial membrane is our electron transport chain. It is a complex of different proteins that is transporting electrons from one complex to the next, with the end result of reducing molecular oxygen to water. At the same time that happens, we're getting an outpumping of protons that accumulate within the matrix of the mitochondria. They get pumped out into this inner membrane space, and they accumulate over top what's called complex 5, or sometimes also known as the ATP synthase complex. And that complex allows for hydrogen to move back through its chambers. It brings together adenosine diphosphate and inorganic phosphate to make ATP. So this is the mechanism in how we make ATP. But notice that cytochrome C sits on the outer edge of the inner mitochondrial membrane. And so things that cause disruption of the mitochondrial membrane, whether it's a toxin, whether it is some sort of structural damage, whether it potentially is an infection, whatever it might be that's damaging the membrane, like polyunsaturated fatty acid oxidation or lipid oxidation, we get an outward expression of cytochrome C. And when we get an outward expression of cytochrome C from the inner mitochondrial membrane, that triggers a series of enzymatic reactions that lead to cell death called apoptosis. And so it turns out that the 4-hydroxynonenal that occurs because of lipid oxidation of the cell membrane or the inner mitochondrial membrane can cause an outward expression of cytochrome C, triggering apoptosis. And so the mitochondria is a very sophisticated organelle. It has basically a number of different components. We have an outer mitochondrial membrane. We have that space that sits between the outer and the inner mitochondrial membrane where the protons accumulate. It has something called cristae, which are these folds of the intermitochondrial membrane that increase the surface area for ATP production. And then inside the matrix is where the Krebs cycle, that's also where our mitochondrial DNA lives. And so much of the activity of the mitochondria occurs along the intermitochondrial membrane. The intermitochondrial membrane is made up of phospholipids and other proteins. Part of the phospholipid content of the intermitochondrial membrane is going to be fatty acids that some of them have more than one double bond. But it's through our diet, and it's through changes that occur over time through the consumption of many vegetable oils, particularly seed oils that are very high in polyunsaturated fatty acids, that change the contour or the content of our phospholipids. And we start moving phospholipid fatty acids to more of the polyunsaturated fatty acid forms, that we increase the potential for these lipid oxidation reactions. And that can happen within the intermitochondrial membrane, just like it can happen to our cell membrane and to the membranes of the other organelles of our cells. Now, it turns out that the intermitochondrial membrane of the mitochondria has a protein to phospholipid ratio of something called 3:1, with a specific phospholipid called cardiolipin. And cardiolipin is unique to the intermitochondrial membrane of the mitochondria. And it too can become damaged because of lipid oxidation. And it's very unique in what that particular phospholipid does in helping with the structural integrity and function of the electron transport chain. I have another mechanisms video that I have recorded that talks about the role of cardiolipin in mitochondrial function. So I would encourage you to watch that video separately to learn more about cardiolipin. For more information like this and a lot more, you can join us in our Mitochondria Mastery course. The course is available through Integrative Medicine Academy, or you can access information through mitochondriamasterycourse.com. You can email us with any questions you have about our courses at integrativemedicineacademy@gmail.com. So once again, I'm Dr. Kurt Wohler. Thank you so much.